and why it is important to screen for neutralizing antibodies. So that kind of webinars initially had, and we also tried to loop in Professor Wang last time, but unfortunately due to his health, he was unable to join us. And I think this is a good time to have both Professor Linfa Wang and Dr. Sengupta here, because both are very pioneer in their each own field. And in India, there are a lot of vaccination strategies are going on, and we have 5 to 6 vaccinations that are going upon, and the government has planned different phases of vaccination. Right, in the phase 1, everyone are where only the frontline workers were targeted, and during the second phases, people who are aged 60 plus were targeted, and during the second phase, people who are above the age of 45 were given the preference. And now during the fourth phase, who are all above 18 can get their vaccination done. So even though there are a lot of vaccination is going upon, we are hearing certain episodes of reinfections, or episodes of some kind of bad news that we are hearing. And there are talks about seroprevalence and serosurveillance studies for after post-vaccination. Even though there are a lot of clinical trial happened globally, still in India we need to understand how the vaccine works, and whether if someone is vaccinated, how long the immunity is it prevails in that individual, and what are the parameters that we need to check to find out whether we are vaccinated or not. Sorry, whether the immunity is developed or not. So it is important for us to ensure that we have a sense of pandemic preparedness moving ahead, and to keep ourselves safe, open up the economy, and provide confidence at the individual level. Even though we are still in the pandemic level, but there is a focus and attention required for seroprevalence, and there are a lot of studies in supporting that antibody seroprevalence is essential for monitoring the COVID pandemic and the vaccine uh, task. So here we have Professor Linfa Wang and Dr. Sengupta. Professor Linfa Wang will be talking more about the importance of neutralizing antibodies in COVID-19 management, and introduce the novel CPAS technology, which is a FDA EUA approved kit. Professor Wang, he's from Duke NUS, Singapore. So he's a professor in the program in emerging infectious diseases. His research focus is on bat-borne viruses to better understand the virus-bat interaction. So he holds a number of honorary positions and memberships, and has received numerous awards, such as 2014 Eureka Prize for research in infectious diseases. It's an honor to have you, Professor Wang, among us. The dice is yours.
Thank you, Mani. And so let's get direct into the slides. And okay, you can see okay, right? Okay, slides okay?
Yes, it is.
Okay.
Yes, it is.
All right, so really this is a very kind of a quick talk on why we need to do that, the post-vaccination surveillance and the neutralizing antibody. Okay, so what is neutralizing antibody and why we need to test, and what's the best way to do it. To set the scene, Manny already mentioned, but this is really now, this time last year, we're all hoping to have a vaccine, and we thought with a vaccine, future will be bright. But a year later, and some areas already, quite good coverage. So now we realize that infection after recovery of vaccine can still happen. So there are two terminologies. Reinfection really describe people recovered from COVID-19 and still get, infected. Unfortunately, very few cases that we have their neutralizing antibody level before reinfection, but with a half a dozen or so we have, uniformly they show low or no neutralizing antibody. So I think the number is not big enough to conclude, but one thing is clear: neutralizing antibody plays an important role to prevent, reinfection. Same with breakthrough infections. I know that India right now, we are really getting off the peak, and but clearly the data shows that even vaccinated people can still get a breakthrough infection. So for a country where you have still a large scale of transmission, that risk is high. But what's surprising us is for what we say the most controlled nations, such as China, Australia, and Singapore. So we don't have, massive transmission in the society, and yet, with the very, very sporadic clusters of infection, it still involves people who already have 2 doses of vaccine. So breakthrough infection can happen in most controlled nations. The last point I'll try to make is, from data from Israel, for example, that is have a very good vaccine coverage. So then you can see clearly that in 2 populations, naive population, you have infections of all sorts of different strains, the alpha, the beta, the delta. But in the vaccinated population, if you're unfortunate enough to get a breakthrough infection, its majority is the beta virus, so associated with the variants of concerns. And then just some basic, immunology, that if you get exposed or vaccinated, your body will working hard to fight against the infection. And there are 2 arms, right? One is the cell mediated, one is the humoral. The humoral will produce the most important antibody, the functional antibody is neutralizing antibody. And on the other arm, you have T cell immunity. So people always ask, which one is more important? And the standard answer is both are important, right? Both neutralizing antibody and T cell plays a role in preventing and controlling diseases. But neutralizing antibody is by far the more important arm in preventing infection and transmission. The reason is that neutralizing antibody can act before the virus even enters the cell, because T cell will only be activated when the virus is already replicating in your body. But there's also a practicality consideration, right? Currently, there's no T cell assays for larger scale testing. And in the immediate future, I don't think the FDA or any country regulatory bodies can approve a T cell assay, because the T cell assay is just much more involving and very hard to do quality assurance. So just one slide of virology and the neutralizing antibodies, right? I mean, you have seen this thousands of times, this computing sort of enhanced image of the COVID-19 virus. And on the section, you can see that the spike protein, triangular protein, is that spike protein. And that's the genome coding for all the different proteins, and the spike protein is here. So when you are exposed or vaccinated, your body will induce all sorts of different antibodies, and they will bind to different parts of the virus and different parts of the protein. What we call them is binding antibodies, BABs. But the functional part is the neutralizing antibody. But neutralizing antibody is very targeted. And through the SARS-1, COVID-1 research that we, fortunately, 17 years of accumulation, we know exactly which protein where this neutralizing antibody targets. So it targets this spike protein, and it's a small proportion in the spike protein. It's called the receptor binding domain, or RBD. Okay. So the gold standard right now to measure neutralizing antibody is what we call virus neutralization test. So you need to have a live virus. You really protect yourself in a biosecurity level 3, BSS3, full PPE protection. The principle is simple, right? The virus will incubate in the cell, and if they find the ACE2 receptor and go in, and then you cause the CPE, kill the cell. And then if you have neutralizing antibodies, hopefully that will be blocked and the cell is protected. So the advantage is that it's highly specific in the function, measure the functional antibody. There's lots of disadvantages. It requires the biosafety containment, very tedious and slow. You need a highly skilled personnel to read plates like that, and very expensive. So, more than 15 months ago, we invented this technology called the Surrogate virus neutralization test. Basically, we try to replace this tedious and unsafe kind of test into a very, very simple, and the paper was published in Nature Biotech July last year. So put it simply, SVNT is about chemical simulation of the live virus neutralization test. So panel A is the live virus neutralization test in diagram form, right? The virus binds to ACE2, and in the presence of neutralizing antibody, you block that. For SVNT, basically, what we did is through protein engineering, we express the soluble part of the receptor so that we can purify and coat onto ELISA plate. And for the virus, we take a very small part, is the receptor binding domain RBD. I show you in the previous slide, the red bar. And we chemically conjugate to the most frequent used reporter enzyme, the host reactive peroxidase, HRP. So now a complicated 3 to 5 day experiment needed BSL-3 was transformed into 1 hour test in any BSL-2 lab, whether it's clinical diagnostic lab or research lab. So the principle is the same. In the absence of neutralizing antibody, RBD will bind ACE2 and develop a color. In the presence of neutralizing antibody, either this is eliminated or reduced. Okay, so when you declare you have a neutralizing antibody test, which is a surrogate, people will ask how good it is to the authentic live virus neutralization test. You know, we have lots of data. I just show you one slide. So this on the x-axis, this is the live virus, the PrEP reduction neutralization test titer, and this is the CPAS titer. And as you can see, the R square is 0.95 close. Very, very good. And then in the paper, we also present data on the specificity and the sensitivity using 2 natural infection cohorts, one in Singapore, one in China. The product was launched on May 15th, and GenScript is the manufacturer, and we are the inventor, and then the DXD hub in Singapore is our collaborator, really in quality assurance and the regulatory approval. And then on November 6th, you know, this is a huge milestone. We got FDA approved as the first and remain the only test that specifically detects neutralizing antibodies against SARS-CoV-2, either from infection or from vaccination. The question now people ask a lot is that, okay, you know, a lot of people, even in the general public, are very used to rapid antibody, rapid antigen test and PCR test, and they're less knowledgeable about antibody tests. So this is the why, you know, why we do an antibody test. Can antibody tests protect immunity? Actually, we have 2 different sub-questions. The first question is that the commercial antibody tests currently on the market, majority of them, you know, 99% of them is only measuring binding antibodies. So can binding antibody assays accurately measure the neutralizing antibody? That's question number 1. Question number 2 is that with kits like CPAS, now we're directly measuring neutralizing antibodies. Can neutralizing antibody be used to correlate protection? So I will address these 2 questions separately. The first question, can binding antibodies, you know, accurately measure neutralizing antibody? No, because binding antibody is not neutralizing antibody. So very early on, when we submit this paper to Nature Biotech, we already had this data from monoclonal antibodies. So these are 4 different monoclonal antibodies selected on their ability to bind this RBD, the receptor binding domain. So it's exactly the same recombinant protein used in ELISA direct binding, and use ELISA for CPAS, which is a neutralization test. So as you can see, 4 different neutralizing antibody, and the best one is this red one, 4A1D10. And then you come across to panel B for neutralization test in CPAS, as you can see. The best binders fail to neutralize. Only this green one, the blue ones, is really a neutralizing antibody. So clearly that binding antibody is not neutralizing, and the best results come from monoclonal. How about human sera? So these are the human sera data, you know, not from us, from other groups. They compare the live virus neutralization titer, which is on the x-axis, and then the binding antibodies on the y-axis. You know, in this very early study, they used the Roche, Abbott, and the Euroimmun. Again, this is third-party data, not our data. You look at the R square, 0.3, 0.4, 0.5, 0.5. Again, I remind you, ours is 0.9. Why there's such a big discrepancy from their test but not our test is because they are measuring 2 different things. The neutralizing antibody, live virus is match only neutralizing antibody, whereas all these serology tests are only measure binding antibody. So they try to correlate with neutralizing antibody rather than measure neutralizing antibody. That's why the correlation is not as good. Second question, once you can accurately measure neutralizing antibody on large scale, can you use that as a predictor of protection? And the answer is yes. And that's not my word. These are the words from Johns Hopkins and from Tony Fauci. Not only their word, we have more and more data now come out from published studies. So I don't have enough time to go through the lit review with you. I just highlight, two papers. One is on a specific study. The other is really on a mathematical simulation and modeling of all the neutralizing antibody data published so far. So the first study is a very neat study, and very early on in July, that the Washington University from Seattle, they started a cohort. 122 people on a fishing vessel go out for fishing. And what they did is to make sure all these people are PCR negative, all right? Because if you're PCR positive, of course, you cannot go out because you can infect other people. To their credit, they also took a blood sample before these people, go out at sea. And unfortunately, obviously, that 122 PCR negative does not mean all of them are clean. At least one of them, we don't know. At least one of them has a virus, maybe during incubation. So PCR negative, but still manage to spread this virus to their colleagues. And the end results is that about a month later, 85% of people on that vessel, 104 out of 122, all came down with COVID-19. Basically, they were infected with an attack rate of 85%, which is not unexpected for COVID-19 if you have a close environment and people have a close contact. I just show you one data from table 1. As I said, before they went out, PCR all negative, but for serology, interestingly, they got 6 positive by the Abbott binding antibody assay. To their credit, they also carry out 3 different neutralization: live virus neutralization, cellular virus neutralization, and CPAS blocking ELISA. As you can see, out of 6 positive on binding antibodies, only 3 are neutralizing positive. The cutoff was 20% at those days, and now it's 30%. So 3 negative, 3 positive. Look at when they come back. The 3 positive neutralizing antibodies are 100% protection. Remember, the attack rate is 85, so it's highly, highly significant. Neutralizing antibody is a predictor of protection from this study. For those who have the binding antibody positive but neutral antibody negative, 3 out of 3 all got infected, and CT is 17 to 22. So they're not only infected, they have very high viral load. So a beautiful study, and clearly demonstrate neutral antibody is a predictor of protection. This is a much more comprehensive study. Basically, they gathered the published data from 7 vaccine trials and also the convalescent cohorts. The key finding is that neutralizing antibody level is highly predictive of immune protection. And look at the data. So here is neutralizing antibody from convalescent plasma, either from vaccination or from infection. So the convalescent plasma data is here from natural infection. And we know the mRNA vaccine and the co-vaccine that you're using, and also the AstraZeneca vaccine, it's all here, right? So the AstraZeneca is here, and then you have the inactive vaccine. So this is the neutralizing antibody gradient from high to low. This is the protection efficacy from high to low. I mean, it's not 100% perfect, but it's as good as you can get. And then they use this data to do a prediction. So this is a predicted efficacy, and this observed efficacy, and now they have a linear plot. Again, these dots are not on the line, but off by not much. So from these two papers, we can conclude the neutralizing antibody is definitely a predictor of protective immunity. The next question is how long this neutralizing antibody will last. I mean, you must have seen papers, newspaper reports, this time last year was very pessimistic to say, oh, neutralizing antibody only lasts 3 months, 4 months max. Now you have papers that say, oh, the neutralizing antibody can last 8 months, at least 9 months. Both are right, both are wrong. They're talking about an average or trend, whereas if you really, from a risk assessment point of view, you want to know your own immune longevity. So we did that. We published that a few months ago in Lancet Microbe. It's a cohort of 200 COVID-19 patients recovered, and 164 was kind enough to coming back for multiple sampling. And the data is a 9 months longitudinal study. The key finding is that the longevity of neutralizing antibodies is very, very diverse. It's not a uniform, okay? It can last from days to decades. So you can read the paper for detail. I just want to present one data slides, is that from the cohort we had in Singapore, we can divide them in 5 different dynamics group in terms of neutralizing antibody. The first group, they never had it. The last group, they gradually increase. 9 months later, their neutralizing antibody is still going up, which we really don't understand. But because the number is small, so we exclude these 2 groups, we call them the outlier. The majority of the people who recovered from COVID-19 is in the middle 3. They had the neutralizing antibody, but then very fast or slow, or some of them really keep for a long time. So how long the neutralizing antibody will last? From this group, the individual, the shortest is 35 days, longest is around 150 days. Median is 100. The second group, double that. The third group is the most heterogeneous. It can last a year or 14,000 days, which is 40 years, okay? So definitely there's a lot of heterogeneity. And now, the other worry, obviously, is the variants of concern. So we now have alpha, beta, gamma, and delta, all right? And we have variants of interest on the horizon as well. The 3 questions people ask the most, are they more transmissible? The answer is clearly yes. Are they more virulent? Yes, for some, the others are less clear. Can the current vaccine protect against them? Yes, if your neutralizing antibody is in the high group. No, if your neutralizing antibody or T-cell immunity is in the lower group. So again, this is a difficult answer. Depends on who you are and where you are with your immune longevity and vaccine response. So this is the data I promised to Dr. Sengupta I'm going to present. Is preliminary, and we are still drafting the paper for this. So this from healthy donors who have been vaccinated with mRNA vaccine. In Singapore, the first vaccine was Pfizer and the second was Moderna. So we sampled this individual N equal to 20, day 0, before they take the first jab. Of course, they are all negative because they are healthy donors. They have never been exposed to COVID-19. We take on day 21, so that's before their second jab. So they had 3 weeks to develop their neutralizing antibodies. And the black is the wild type strand, the blue is alpha strand, and the green is the beta. So the blue, look at that, only 1 amino acid change in the RBD domain, and we're already seeing a difference. And the green, you have 3 different amino acid changes, certainly is they have neutralizing antibody, but they're lower. Okay, so this is the first injection. Day 35, that's 2 weeks after your second injection. For the wild type virus, as we all know, the mRNA vaccine really induces high neutralizing antibody, and it's almost uniform. The protection is very, very high, and almost saturation, close to 97, 98%. But for the alpha variant, it's not so, and for the beta, it's even worse. 90 days now, just over 2 months after the 2 doses of vaccination. Now you look, even the wild type virus, you start to see the spread of the point. Alpha, yes, and the beta is the most worrying because that individual is now close to 50% inhibition. Remember, 30% is our cutoff, so it's very close to the cutoff. It only took 2 months for that individual. The other point is very important. Again, binding antibody, you cannot appreciate that. So we took the sera from that group at the peak 2 weeks after second dose, and we conduct a binding antibody on 3 different IBDs, okay? So the color coding is the same. Black is the wild type IBD, the blue is beta, so alpha, and the green is beta. So you compare this with this. Now, look at that. If you measure a binding antibodies using any kits, even if you included the variant IBD, you could not differentiate because the binding antibody is masking the difference in neutral antibody. So you look at this and this. If you did not do this, you think, oh, no problem. Our antibody is very high. No, it's high against the wild type, but already low against the different variants. Okay, so this is just more variants. We have alpha, beta, and we put SARS-1 into it, and we have the bat, rat G13, and the pangolin virus. I call this the potential SARS-3 and the SARS-4 virus in future. So these are SARS patients. They only neutralize SARS virus, and some of them have very weak neutralization against the other variants. And these are the COVID-19 patients. We know the wild type is the best, and then they come down like this. And after vaccination, it goes up. So this is day 35, right? But I already can tell you that, by the time you have 2 months, 3 months, 6 months, 12 months, these guys will look like that. They will come down, okay? So factors to consider for immunity passport. We all want to get out, right? We want to travel domestically, internationally, and there's talking about, how we can open borders. And one kind of area to consider is immunity or vaccine passport. There are many factors we have to. One is protect immunity. We're still debating about whether it's neutralizing antibody or T-cell immunity. And I said, you can debate forever for science, but on the practicality level, you can only measure neutral antibody on large scale with CPAS, and you cannot do that with anything else. And vaccine efficacy, are we talking about against disease or transmission? Immunity longevity. Now we have so many different vaccines, right? You have different vaccines against the same virus, or you have the same vaccine against different variants. So the whole picture is very, very complicated. Very early on, the Australian Qantas airline was saying, we want to really implant a vaccine passport. And so that for international passengers, at least you have to say yes to the question of, are you vaccinated? And I say, that's not enough. Maybe we should ask people, are you CPAS? Because vaccination does not equal to immunity or protect immunity. Okay, so with that, I'm going to just stop here and thank you for your attention.
Thanks a lot, Professor Wang. I think you have now summarized extensively about what is the importance of neutralizing antibody and how it is important. Even though someone is vaccinated, you can't be sure that you are truly immune to that, from the infection. And the recent data that showed that, even though many people are looking after only the binding antibodies, and binding antibody is not in the true marker to look for immunity against the infection. With that, I think we can take the questions at the end of the session. Let's go to the next presenter. We have Professor Shantanu Sengupta. I think it's a delight to have you, sir, here. And Dr. Shantanu Sengupta is a senior principal scientist from IB. He works mostly on the clinical genomics, epigenomics, and proteomics. He's a PhD from Csr IICB, and he has done his postdoc from NII and the Cleveland Clinic Foundation. Most of his works are towards understanding the genetic and proteome and the metabolic basis of cardiovascular diseases, and is a counselor member of multiple societies in India and the International Society of Heart Research. And he is an honorable editor in Journal of Genetics and academic editor for PLS1. Today he will be talking more about his experience with the CPAS and implementing the ongoing COVID-19 vaccination drive and relevance of screening utilizing antibody in a post-vaccination surveillance. Welcome, Dr. Shantanu Sengupta.
Thank you, Mani. Let me thank Premas for inviting me here. And I would like to thank Professor Wang for giving a wonderful talk explaining how CPAS is important. So what I'll do in the next 20 minutes or so, oh my God, there is some problem in slides moving. Can I stop here and reshare? Sorry
yes,
about
no
it.
issues.
Okay, so what I'll do today is I'll talk about two different studies that we are doing currently, and we have extensively used neutralizing antibody and what advantages did that give us. I'll just try to explain to the audience. So let's come to the first study, that is the study what we call Csr Cohort. So what we have done here is we have developed, and this is, a gift of COVID, that we were able to convince all the people in CSIR, and we have created a cohort of CSIR people, and you can see all these, the CSIR laboratories are actually present all over the country. So this, with a pan-India presence, we wanted to understand the burden of COVID-19, and also because it was a longitudinal study involving CSIR personnel and their family members, we were also able to see the stability of the antibodies. So basically what did we ask? We asked, do you have antibodies against COVID-19? And if you have antibodies, how long will they last? So this was a slide that we prepared in June 2020, and we started the exercise from IGIB. I remember it was 16th of June 2020. So we started, the first sample was collected 16th of June 2020. So it's just 1 year. And we have now done 3 rounds of collection. The phase 1, although it started in June in IGIB, but majority of the country, it was done between August and September. The phase 2, we have done February and March. And phase 3, we are currently doing, and we expect to finish it by July. So you can see here, we had a collection just before the wave, the current wave that we have, the India has experienced, and we are now going to do it just after that, and this will give us a very nice handle to see the effect of the current pandemic. I don't know what is happening. Suddenly, slide stopping. Okay. So if we go to the first phase of sero survey, so you can see here that there was different kind of seropositivity, and this I'm talking about binding antibodies. So this was actually a qualitative assay against the nucleocapsid protein of coronavirus. So you can see regions marked in green, red, and yellow. Red were those regions which had less than 5% of seropositivity. Yellow were those regions which had seropositivity of 5 to 10%, and green were those regions which had more than 10% seropositivity in August, September. So we also then looked at how the disease spread, and if you can see, we divided the seropositive so what we did was we divided the population, classified it based on population density. So low density were areas where the population were less than 1,000 per square kilometer. Medium density was where population was between 1,000 and 10,000. And high density areas like in Delhi, Hyderabad, Bangalore, the number of people were 10,000 people per square kilometer. So if you can see in the slide that high density areas like the metropolitan metros that we call, they had more than 50%, 15% seropositivity in August, September, while the low density areas had almost 5% seropositivity. And the most important thing was that and this is probably the first time we showed that, although it was intuitive, that wherever the seropositivity was greater than 10%, the test positivity rate in the next 30 days actually declined. What it means that number of cases divided by the number of tests done, RT-PCR tests done, declined in places where the seropositivity was more than 10%, whereas it increased in places where the seropositivity was less than 10%. So this can be an interesting handle in future for the government to understand where in the next 30 days the progress can, the disease spread will happen more and where the disease spread will happen less, and accordingly manage the resources. So we also found, and this was interesting, that 75% individuals were asymptomatic, which means that these people probably never they got themselves tested because they didn't have symptoms. So the cases that we are talking about is probably much lower than the actual burden of the disease. And this can only be done using a serosurvey where you can probably accurately determine the actual burden of the disease in a population. And amongst the people who had disease, this is a no-brainer. Fever was the most common symptom, followed by tiredness, cough, and so on. We then wanted to see, okay, these people have antibodies, but do they also have neutralizing antibody? And we used the CPAS kit. And what we showed here was, at that time, the CPAS neutralization was 20%. So anything above 20% was having neutralizing antibodies. So we found that at least 95% of the people who had seropositivity also had neutralizing antibody. Although, please remember, we are not talking in terms of quantitative terms, but we are saying that people with antibodies against nucleocapsid protein also had neutralizing antibodies. So only 5% of them did not have it. Now, this slide I need not show because Professor Wang very nicely explained, but this is a paper published in 2020, which clearly shows the correlation between your CPAS neutralizing antibody and the PRNT50 assay, and you see a very nice correlation. So although they had taken 20% as cutoff at that time, but subsequently the CPAS has become 30%, and you could see 30%, if you take a cutoff of 30% in this, then even the false negativity is very low. So what we then did, and I understand we didn't look at individual antibodies. So what we did, individual people for their, what we did was we tried to understand the stability of the antibodies. And it is similar to what just Professor Wang said. So if you look at the left, so if you look at the left-hand panel, this is the x-axis is antibody levels at baseline. That is the first time when we collected. And since this was a longitudinal study, we could follow these patients with the samples for multiple time points. And the y-axis is the antibody levels at 3 months. So you can see in most of the cases, the antibodies are to the top of the dotted line, which indicates that the antibody levels actually increased. But now let us look at the neutralizing antibody. The neutralizing antibody, on the other hand, actually started decreasing. And in 5% of them, you did not have neutralizing antibody if we take 20% as the cutoff. What happens 5 to 6 months later? So 5 to 6 months, here also you can see that in a lot of people, the antibodies levels increased, but in some of them, it started decreasing. But look at the neutralizing antibodies. In some of them, yes, as Professor Wang pointed out, some of them there was an increase, but most of them now it decreased, and we found that about 20% of the people did not have any neutralizing antibody after 5 to 6 months. And this is the 3 months is based on about 600 samples, and the 5 to 6 months is based on about 160 samples. So you can see that although you have antibodies, you have actually lost the neutralizing antibodies. And this result we got has now been published. And this is the result that we got in about December, January, when we started doing the second phase. As I said, I told you we started the second phase in February, March, but in IGIV we started collecting in January. So if you look at the data February, March, so if you look at this was August, September, where the seropositivity was 10% overall. Come February, March, the seropositivity increased to 28%. And interestingly, in Delhi, we found that the seropositivity was 42%. During the same time, Delhi government also conducted sero survey, and they mentioned that the seropositivity was around 55 to 60%, which is possible because their sero survey is mostly based in slums, whereas and so it will be relatively higher than average. And our sero survey is based on CSIR labs, and because it's a scientific institution, the awareness amongst people are a bit higher. So we will expect our seropositivity to be a little bit low. So if you take an average, it's about 49%. So now let's look at Delhi. So the question was, even when we had such high seropositivity, why did we see such a catastrophe in April? So to address the question, let us understand this. So this was Delhi, and we broke down Delhi into different regions based on the institutes from where we collected the sample. And if you see that wherever there was a laboratory setting, like in IGIB, it was 43%, where it was just public dealing, where it's mainly in the offices, it was 49%. And in residential areas, so CSIR has residential colonies, it was much lower, 28%, and an average was 42%. So with such high seropositivity, why did Delhi see such a bad situation during April, May? To understand that, we looked at the quantitative antibodies this time, and these were antibodies from Roche. So you can see that in phase 1, and mind it, this is log scale. So the red line here shows that this is what is recommended for a convalescent plasma. That is a plasma, the antibody levels for this kit should be at least this much to give you certain protection. And you can see that even in phase 2, that is January, February, March, you can see that about 42% people had antibodies. Of these, more than 50% did not have antibody levels sufficient enough to be considered to be effectively protected. And if you now look at the right-hand panel, where we correlated your quantitative binding antibody levels with the CPAS antibody kit. And you can see that even though people had antibodies, about 20% of them did not have neutralizing antibody. So although the seropositivity was 42%, the effective seropositivity could be much lower, because 20% of these 42% did not have a neutralizing antibody. And if we consider that we want to go above 50% neutralizing to be more confident, so there were only very few people who were actually having high neutralizing antibodies as well as high antibody levels. So what we did was, during this time, since we were doing the study in IGIB, we had followed up at least 60 people, at least 75 people serially, and we had data for them for at least 3 time points. So in July, we wanted to see how many of them, so 65 of them were previously infected. So how many of these 65 people were actually reinfected? And how do we get that? We look at the N antibody level against nucleocapsid antibody at various time points. So initially there will be a dip. So if there is a dip and then sudden rise, those were the cases which were reinfection. And in this case, we can see about 14% reinfection was reported. As I told you earlier, 19% of the people did not have neutralizing antibody, and we see from our data in IGIB, and this is I understand this is a small sample, but if you look at the data, and we hope to get large number